taken your theory class with the third batch or with the first or second batch on Monday?
Third batch, sir, I think.
Monday तो practical था ना sir उस दिन?
So
No,
practical
Sir, I
with
guess
was
whom
first
are?
for
batch
third
was
That
with
batch.
was
all of
I
us.
think
Okay, so
third
I
batch
have
का
to
From
practical
all of
था
us.
अपने.
no, I have to start from the beginning now for this class. Or I have already discussed something with you in theory class.
सर
So
थोड़ा
you
बहुत
have
बताया था
already
सर आपने
Okay,
brief
कैरेक्टरिस्टिक्स
overview.
आपने
अच्छा,
okay, okay, okay.
कैरेक्टरिस्टिक्स के बारे में बोला था शायद.
Okay. Let me explain. It will take a few minutes if we are revising it. So this is diversity of coordinates paper, and there are some objectives which we have fixed, keeping in mind while going through this paper. So scope and historical background of coordinates we will be discussing. Then origin of coordinates and characteristics and classification of chordates and the exclusive phenomena which are seen exclusively in chordates, higher chordates, like mechanism, biting mechanism, snakes, flight adaptation, and some physiological adaptations and mechanisms which are exclusive to chordates uh, are kept in objectives. And after going through this entire paper, there are some learning outcomes which are supposed uh, to be learned by the students. So they must be knowing about the habit and habitat of chordates in marine, freshwater, and terrestrial ecosystems. And the students should understand different classes of chordates, level of organization, evolutionary relationship between and different subphyla and classes, and within and outside the phylum. And they should study about diversity of chordate and study of various systems of chordates. Then this is about the syllabus. So you will get 4 credits for successfully completing this paper, and there are 60 are given for teaching this paper. So there are 10 units. So first 6 units I will be taking, and rest 4 units will be taken by Mohit sir, and these are the recommended books for this paper. So the classification we have to follow by the Young's JJ 2004 third edition, and there are some other books like Parker's and Poe. You can also go through it. You can also refer these books for your knowledge enhancement. And about the syllabus of the practicals, so you will get 2 credits for completing this paper. And mostly there are museum specimens for practicals, because practically we do not dissect an animal. Nowadays we only study the preserved specimens, and while studying the preserved specimens we get to know the morphological, anatomical, and physiological characteristics, as well as we must see some videos of the animals living in their who are actually living in their natural habitat. So we get to know and get to see how they behave, how they do different physiological things in their natural habitat. And besides these museum specimens, there are some sections as well, so that we should understand the histological pattern or histological studies of the given organs and their specific information. Then we have some animals for to be assigned to the student to know more about these animals. So there are 2 animals which will be assigned to each student. They have to make a project on them. And there is another thing, that is Weberian ossicle of Mistus. So we will be studying these ossicles by observing and studying the photographs as well as some videos and their importance for this fish. As in the previous class I had told you that these Weberian ossicles Weberian ossicles remain connected with the swim bladder, and they enhances or amplifies the sound waves, so they add in hearing for the fish. And the pecten from fowl, or you can say the pecten, is a structure which is found inside the eyeball of birds, and that provides nourishment to the eye, and it also absorbs the ultraviolet rays, so it sharpens the eyesight of the birds. So now coming to the chordates, the introduction, because this is our first unit, introduction of chordates. So we should know that why some animals are called chordates. So first see meaning of the word chordata. So the chordata word derived from two Greek words. First is chorde means string, and second is eta means bearing. So these animals must be bearing a cord at their back. So this provides them mechanical support, and this also help in making a perfect structure of the body. Then the notochord, which is one of the characteristic of all the chordates, that also derived from two words. The first word belongs to Greek, that is noton, that means back, and the second word is chorda, that means cord. So these animals again bear a cord at their back, so that supports their body. It provides the mechanical support. So with the help of this cord, they bear the load of their body, and it also helps in forming their body shape. And if we see the basic characteristics of the chordates, so there are three basic fundamental characteristics of chordates. The first one is dorsal tubular. First one is the notochord, presence of notochord. So you can see in this diagram, this is notochord. This is a rod-like structure that is running or extending from anterior end to the posterior end. And the second thing that is found above the notochord is dorsal tubular nerve cord. So this is the second basic characteristic. So whenever the nerve cord is found in chordates, it should be hollow, because we know that nerve cord is also found in non-chordates, but it is not hollow. So this not hollow space of the nerve cord remains filled with the white and gray matter. So that actually is made of different stuff or composition of nerves, and you can say neurons. And the third basic characteristics is the pharyngeal gill slits. So the pharyngeal gill slits are found in the pharynx region. So pharynx is a part of the alimentary canal. So these are the slits by which the pharynx opens to exterior, and these slits are highly vascularized. They are supplied with blood capillaries. And because of their vascular nature, the gases are diffused. The gas exchange takes place in the walls of these gill slits, so they help in respiration. And the another feature, these are the three basic or you can say fundamental features of chordates. On the basis of that, we can say the particular animal belongs to group chordata. So the first one, the notochord, it is longitudinal cartilaginous rod running between the nerve cord and the digestive tract. And the second characteristic of notochord is it acts as a support for the nerve cord, and it is replaced by the vertebral column after the embryonic stage in all vertebrates. And second uh, feature of the chordate or the basic characteristic, dorsal nerve cord, it is a bundle of nerves running along the back and splits into the brain and the spinal cord, and it is hollow and lies dorsal to the notochord. The third characteristic, which I explained a little while ago, pharyngeal slits, they are the opening which allow the entry of water through the mouth without entering the digestive system. So all chordates have these openings on the lateral side of the pharynx at some stage of their life. So that means these three characteristics, notochord, nerve cord, dorsal nerve cord, and pharyngeal gill slits should be there at some stage of the life cycle of an animal, if we consider that animal in group chordata. The fourth point, which is not fundamental, which is not basic to all the chordates, that is post anal tail. So it is an extension of the body to the anus. In chordates, the tail is composed of skeletal muscles which help in locomotion in fish-like species. It is absent in most of the adult chordates. So the post-anal tail is not universal. It is not fundamental to all the chordates. It may be found. It may not be found. But as we just came to know, we got to know that skeletal muscles are also attached with the tail. So you can see these are the muscular post anal. This is the post anal tail, and these are the muscle segments which are called myotomes. So we know that these myotomes help in locomotion, and they also help in movement of the skeleton. So these are also important features of the chordates. So besides three basic characteristics and one post-anal tail feature, there are some other characteristics of the chordates which are unique, which are not found in non-chordates, or if they are found in different forms. So the first feature is axiation. That means the distinct polar axis. So the chordates have anterior and posterior axis. They have dorsal and ventral axis, whereas we know that there are some the almost all the non-chordates do not have such distinct polar axis. So this is the another characteristics which is unique to the chordates. Then the bilateral symmetry, we know that this is not unique, but all the chordates are bilaterally symmetrical, whereas the non-chordates may be bilaterally symmetrical or may not be. Third one is the triploblastic condition. This is equally true for non-chordates as well. Uh, there are three germ layers: ectoderm, mesoderm, endoderm, and the different parts of the body, different systems of the body derived from different germ layers: ectoderm, mesoderm, and endoderm. Say, for instance, our skeletal system derived from mesoderm, whereas the ectoderm derives the skin, the you can say the integumentary system of the chordates. So coelom, the chordates are coelomic. Obviously, the non-chordates are also coelomic, but they may be acoelomic, pseudocoelomic, and coelomic. And if they are coelomic, they are schizocoelomic. Whereas from kynodermates onwards, uh, protochordates, chordates, and echinodermates are coelomic, which are antrocoelomic, because they are, the coelom derived from the enteronic pouches of the archenteron. Then metamerism. So the true metamerism is seen in chordates. They are metamerically segmented in chordates, higher chordates like primates or like humans. The metamerism is not seen externally, but it is very much visible internally. And by the metamerism, we see a high grade of division of labor. So this is important feature for chordate. The next one is the organ system of organization, and we know the high grade of organ system, or you can say the top grade of organ system is seen in chordates, because they bear a very complex kind of body, so they need top class of organ system within their body. Then they have living endoskeleton. Their endoskeleton is made of cartilaginous and bones, and the endoskeleton is internal framework bones. So these bones are not dead. They keep on healing. They keep on rising. They keep on maintaining themselves. But we know that at a certain age, the development of the bone, or the length of the bone come to a standstill. It does not grow further, but it has great tendency of healing itself. So whenever we have any injury within the bones, it has a tendency, it has power to heal up automatically. So we know that the bones are formed by osteoblasts, whereas the cartilaginous are formed by chondroblast. So these are the cells which are mother cells, which make new bones and new cartilaginous. So they keep depositing different matrix, like chondrin protein is responsible for behind the formation of the cartilages, whereas the osin protein is responsible for making the new bones. And we know that in the long bones there is a space cavity that is called that remains filled with the marrow, that is bone marrow, where the blood capsules are made, the RBCs are made. And the long bones also have a unique system, that is Havison canal system, that is a system of blood vessels and nerves. So with the help of these, the animal, the bones get nourishment, and we also feel pain when we get any injury in the bones. So this is the importance of endoskeleton, because it makes the perfect shape of the body. It helps in locomotion, because the skeleton is also attached with the muscles, and it also protects our internal delicate organs from external jerks or some injuries. Then efficient respiration, efficient circulation, and centralized nervous system. So we know that we have pair of uh, lungs for respiration, and we get deoxygenated blood, and then the blood is the air is, I mean, the oxygen is added with the blood, and that it is sent back to the heart, and from there the aerated or the oxygenated blood is again circulated to all the parts of the body. So we have a closed blood vascular system, consists of veins, arteries, and blood capillaries, and we have a circulatory system, closed circulatory system, four chambered heart, and efficient heart, muscular heart. We do not have neurogenic heart. We have myogenic heart that keeps by beating throughout our life, and we have centralized nervous system. We there are three types of nervous system we have, and it is highly developed form of nervous system. And now I would like to show you one video that explains how the
Of all the
chordates.
sea life stalked by Anomalocaris, Pikaia, a tiny worm-like creature.
Okay, so the sound is breaking, so I would like to show you it from different source.
Of all the sea life stalked by Anomalocaris, Pikaia, a tiny worm-like creature, somehow escaped extinction. This bit of an animal had no protective shell or defensive spines. But in the Cambrian
Is the
period,
sound of
one
video
part
coming
of
to
its
you?
design was wholly unique.
Yes, sir.
Yes, sir.
Yes, sir.
Sir.
Yes, sir. Pikaia has a structure remarkably similar to a modern-day animal called Amphioxus. Both are equipped with a long, stiff rod running the length of the body, the notochord. The notochord, which supported Picaia's muscle structure, is the design common to all vertebrates. This modest creature may be our earliest recognizable ancestor.
Pikaia, I believe, evolved into something like a fish, and from the fish, of course, we have the amphibians, represented today by the frogs and the salamanders, for instance, which invaded land in the Devonian. And then after that, we have the reptiles and the mammals. So perhaps if Pikaia had not survived, if it had gone extinct at some time, then of course ultimately there would have been no mammals and of course no humans.
Though much about our lineage remains a mystery, the origins of the human backbone are linked to Pikaia and its notochord. Few developments would lead to greater diversity. The reign of Anomalocaris was not short-lived. It flourished for nearly 20 million years, 100 times longer than the entire history of Homo sapiens. But in the evolutionary lottery, it was the lowly Pikaia that survived, bearing offspring that would eventually populate the seas, the land, and the skies. California's Death Valley, one of the most arid regions in the world. 400 million years ago, an enormous river filled this valley, linking it to the distant sea. It was here that the fossils of the first fish to brave the freshwater habitat were found. It had taken them 60 million years to gain access to these estuaries. The man who discovered the fossil was Dr. David Elliott of Northern Arizona University, a specialist on ancient fish.
The rocks have been slid.
Tons of earth and sand formed sediments at this site. deposited by the river that ran through Death Valley eons ago. And it is here that Dr. Elliott has been focusing his excavation and research work on ancient freshwater pioneers.
Can't really tell what that is. This is a pteraspid. This is a lingulid. And lingulids are rather interesting because they're characteristic of estuarine environments, that is, environments in which freshwater and marine waters are mingling. These are areas of very high nutrients, where you may have large numbers of organisms coming to feed, and we have many pteraspids here. There are also areas that many marine organisms can't get into. They're not able to survive in brackish conditions, and so it may be that pteraspids were protected from some of their possible predators by living in these environments.
Freshwater habitats have almost no salt content. A change in salinity can spell instant death for unacclimated marine life. This paramecium thrives in the salty medium of seawater. Breathing saline fluid, it regulates its mineral levels and ejects wastes. Salt-free water is added, changing the balance. In this unfamiliar, hostile freshwater environment, the paramecium takes in water without voiding it, destroying the cell. The now bloated paramecium ruptures and dies. Pteraspis is about 8 inches long, its head covered with tough bony armor and its body with primitive scales. Unlike paramecium, Pteraspis managed to adjust to this new freshwater habitat with a little creative bioengineering. The pteraspid's protective shell prevented water from penetrating the body through its outer layers. But the gills it used to breathe were a weak spot. To balance its system, developed a chemistry. Transforming its physiology was a major evolutionary breakthrough for the first freshwater fish, but it was only a first step. For 400 million years ago, life on the river was unfolding. But that posed new challenges, rapid currents and waterfalls to negotiate. It took millions of years for marine life to adapt to these new ecosystems. To survive here, Earth's first fish would experiment with many anatomical designs. Miguasha Park lies on northeastern Canada's Gaspé Peninsula. "400 million years ago, this was a tropical region, situated directly under the equator. Etched in this meandering stretch of cliffs is the 20-million-year history of fish evolution. Fossils of freshwater pioneers. Fish with fins to resist river current. Carnivorous fish with jaws and teeth.
Evenly distributed. There's some layers with more species.
And a peculiar fish entirely different from any of the others. This fish had a backbone. Until now, there were fish with tough exoskeletons, but none with hard bone inside the body. Cheirolepis, the earliest creature with a spine, appeared 390 million years ago. The most common fish today, the bony fishes, are its direct descendants. Like modern fish, Charolepis was equipped with one pair each of pelvic and pectoral fins, with sharp teeth for catching prey. Acquiring a spine, Charolepis developed strong muscles to swim with speed and power. But there were other agile swimmers in the river that had managed to do without one. Why then did the backbone develop? While the sea was rich in minerals essential to life, the river was in Working in unison, these cells allow the heart to pump blood throughout the body. To function properly, the heart needs calcium. Calcium concentration in a river is in perpetual flux. When levels are high, calcium is stored as bone. When they're low, the bone reservoir supplies the necessary mineral, regardless of external conditions. Meanwhile, the seemingly solid bone is constantly being replenished. At the top of the screen, hard bone is visible. Moving below are cells that dissolve it. Once the bone is dissolved, calcium circulates through the bloodstream to every part of the body. The fate of ancient fish rests on a simple backbone. Those without eventually perished from calcium deficiency or returned to their native sea. For many years, Charolepis reigned as the king of freshwater fish. His anatomy gained him independence from the marine world. Today, the descendants of Charolepis thrive in both fresh and saltwater habitats. The river was a proving ground for the earliest fish and their offspring. And in this new habitat, they gradually adapted the body system's key to survival on land. Another fish with a backbone appeared on the river at about the time of Charolepis. It was called Eusthenopteron, a bottom dweller. Eusthenopteron was not an efficient swimmer like many fish, but it had secured an evolutionary niche with powerful bony fins to push its way through the river's undergrowth. This fish was a stealthy hunter poised for action. Eusthenopteron's habitat shaped its unusual bone structure, which would one day lead to the evolution of terrestrial animals. South America's Amazon breeds acres of jungle swampland, mimicking conditions in the age of Eusthenopteron. And with the swamps, a host of unusual life forms. Among them, the lungfish, a mud-dwelling creature. This fish has gills, but it also has lungs for breathing air. In such murky water, oxygen content is often very low. The lungfish probably developed lungs to breathe oxygen from the air when the supply in the water was depleted. Just as his ancestor Eusthenopteron had done eons before. Poking its head above the surface for a life-sustaining breath, Eusthenopteron surely caught a glimpse of the world above the waterline. Now that fish had evolved kidneys, bone, and lungs, land was almost within reach. The first four-legged creature appeared 10 million years after Eusthenopteron. Its remains discovered in a forest in Scotland. At the time, this region resembled today's Amazonian swampland. Dr. Per Erik Ahlberg of the British Natural History Museum has been studying the earliest tetrapods. He's trying to find out how and why limbs evolved from fins. Only 10 fossil pieces have been discovered to date. But even from these few specimens, an image of this new creature is emerging. Almost 5 feet long, it looks like today's giant salamander.
I suspect that the reason why limbs evolved was because these animals were living in very shallow water, and limbs can be rather better than fins for moving in those sort of conditions, even if you're not really moving onto land. These earliest tetrapods with limbs rather than paired fins still have gills, tail fins, and other features which suggest that they were very much aquatic animals. So I don't really believe that they were using the legs for walking purposefully over land. They might have made short excursions over land, but more probably they were using those limbs in shallow water.
Today's giant salamander still spends most of its time in the water. Negotiating the shallows is a matter of walking, not swimming. Even in swift currents, the salamander plants its feet on the river bottom and moves around with ease, aided by its natural buoyancy. To move as fluidly on land, the salamander would require a far sturdier skeletal structure. "360 million years ago, the once barren Earth lay under a canopy of green. Giant forests thrived, and small insects now burrowed in the shady wetlands and soil. This landscape was ready to support new life. But to take their first step on land, our ancestors had one more obstacle to overcome: gravity. This is Ichthyostega, an animal that surfaced 10 million years after the earliest four-legged creatures. Judging from the size of his skull, Ichthyostega measured nearly 4 feet long, its backbone and hind legs clearly defined. But this animal also had thick finger bones and a sturdy skeletal structure. Scientists conclude this was the first creature to walk on land. For the first time, an animal with a supporting skeleton around its backbone. The rib cage protected vital organs like the heart and lungs. On land, without the buoyancy of water to cushion them, these organs would be crushed by the animal's weight. By developing ribs, Ichthyostega emerged in a body that could withstand Earth's gravity and move about freely over land. At long last, the time had come to look beyond the water's edge. A 100 million years had passed since the earliest fish ventured from the ocean into fresh water. A habitat that helped shape the complex body systems necessary for life on land. Bolstered by millions of years of evolutionary advancement, Ichthyostega took its first steps while an ocean of life swam in its bones. From sea to river and from river to land, life had taken leaps of astonishing complexity. There is no way to know what called Ichthyostega from the relative comfort of the river toward the challenges that awaited on land. But in taking its first steps, Ichthyostega blazed a new trail, a path we too would forge on our own evolutionary journey from the nurturing seas into the great unknown.
Hello.
Yes, sir.
Yes, sir.
Yes,
So
sir.
I
Yes.
hope
Yes,
Yes.
sir.
so I hope this after watching this video you must have developed some new insight about the life on Earth, how the life
Yes.
evolved on Earth, okay, and
Yes, sir.
especially how did the chordates appeared or invaded on land. I think this video was would have been must have been informative to you, isn't it?
Yes, sir.
Yes, sir. It was very good.
Okay, so now I end this meeting. I have taken your
Okay, sir.
20 minutes
Thanks, sir.
extra,
सर हमारी 4:40 वाली
It's
क्लास
okay, sir.
होगी.
and now I am downloading the
होगी
attendance as
होगी
well.
तो होगी.
हाँ होगी
हाँ
सर
So see,
उसमें भी सबको आना है.
there are
हाँ
there are
सबको आना है
33 people right now.
Okay, sir.
While when I am downloading the attendance, earlier there were 43. So no, there were few more people in the meeting, but some of them left in between. Okay.
thank you, sir.
So okay, thank you, thank you.
Thank
Thank you,
Thank you,
sir.
sir.
Okay,
Thank you,
thank you,
sir.
sir.